This video is for The Living Earth, Unit 4.3, Transcription and Translation. Today's essential question is, what are the causes, types, and effects of mutations? Let's talk about something that's constantly happening in our cells, mutations. Now, when we hear the word mutation, we often think of science fiction, maybe superheroes or some weird creature. But in real biology, a mutation is simply a change in the DNA sequence. That's it. It can be as small as one letter in your genetic instruction book being swapped out. What's really important to understand is that mutations happen all the time, and they're not always bad. In fact, they can be beneficial, neutral, but also harmful. And we'll look at examples of all three. Mutations are a big deal in evolution and in the study of diseases. They explain how species change over time and also why some people inherit genetic conditions.
In all the copying of the thousands and thousands of base pairs of nucleotides during cell division, it is possible for errors to occur. When these types of mutations occur, serious problems can result. Cystic fibrosis occurs when a certain part of the gene is incorrect and the protein misfolds. Down syndrome is the result of an extra copy of chromosome 21, resulting in a total of 47 chromosomes rather than 46. Sickle cell anemia is a genetic disorder brought about by a one-letter misspelling of a base pair in a gene that results in defective red blood cells. Cells have ways of correcting errors. A type of proofreading function scans the DNA and corrects any base pairing mismatches by removing the improper nucleotide and replacing it with a proper one. Some mutations occur spontaneously, but others can occur as a result of exposure to physical or chemical agents. Cancer is a mutation of the cell mechanism that regulates cell growth. It can be caused by such things as cigarette smoke, ultraviolet rays from the sun, exposure to asbestos, medical x-rays, and other natural and environmental causes. Once a cell has become cancerous, it can divide, repeating the cancer in each new cell that is created, which spreads the disease. But not all mutations are destructive. Over the course of evolutionary time, mutations in the cells of organisms have led to genetic diversity and the rise of new and more competitive species, better adapted to their environment.
When scientists talk about mutations, they usually break them into two big groups: gene mutations and chromosomal mutations. Gene mutations are small. They affect only one gene, kind of like changing a word in a sentence. This might not seem like much, but even one letter change in your DNA can have a big effect on your body. These usually happen when cells are copying their DNA, like when you grow or heal a cut. Then there are chromosomal mutations, which are larger scale mutations. Think of these like rearranging entire chapters within a book. These affect big sections of DNA and can mess with lots of genes all at once. These kinds of mutations usually happen when cells divide and the chromosomes get copied or separated incorrectly. So let's zoom in a bit and take a look at the types of gene mutations. These are all about small changes, usually at the level of just one nucleotide base in the DNA. First, let's look at substitution. This is when one base is swapped out for another. It's like typing the wrong letter in a word. Sometimes this does nothing at all, because the genetic code is a little bit flexible. Other times, it changes an amino acid in a protein, and that can change the way the protein works, or even stop it from working entirely. Then there's insertion. That's when an extra base gets added. This is a big deal because it shifts the entire reading frame of the gene. Imagine trying to read a sentence where every word was suddenly jumbled because you added a random letter in the middle. That's what happens in the protein. Deletion is similar, but it removes a nucleotide base. And again, if it's not in a group of 3, it shifts the reading frame and changes everything downstream. These are called frame shift mutations, and they're usually pretty damaging. Now let's talk about more gene mutations. We have translocation. Translocation happens when a piece of one chromosome breaks off and attaches to a completely different chromosome. This kind of mix-up can lead to certain types of cancer, like leukemia. There's also an inversion, which a segment of chromosome breaks off and flips around backward before reattaching. This can mess up how genes are read and regulated. And then we have aneuploidy. This is when there's the wrong number of chromosomes. The most well-known example of this is Down syndrome, caused by having an extra copy of chromosome 21. That's 3 copies instead of the usual 2. Chromosomal mutations don't usually go unnoticed because they affect many genes, and they often lead to developmental disorders or serious diseases. Understanding them helps doctors diagnose conditions and researchers find possible treatments. To make all of this more real, let's look at some specific examples of gene mutations. Sickle cell anemia is caused by a single substitution in the gene for hemoglobin, which is the protein that carries oxygen in your red blood cells. Just one change in one base makes the red blood cells turn from donut-shaped into crescent or sickle-shaped. This causes blockages in blood flow and pain. Cystic fibrosis is often caused by a deletion of just 3 bases in a gene called CFTR. This tiny deletion leads to a protein that doesn't work properly, causing thick mucus to build up in the lungs and other organs. Then there's Huntington's disease, which is caused by an insertion, a repeating sequence that gets copied too many times. Over time, that builds up and damages brain cells. These show how different types of mutations can affect health in very different ways. Now, let's ask, why do these mutations happen? Some mutations occur spontaneously. Remember, DNA is constantly being copied, and even though the enzymes that do this are really good at their job, they're not perfect. Occasionally, they do make a mistake, like a typo in an essay. Then there are environmental factors. These are things from the outside world that can damage DNA. For example, UV light from the sun can create bonds between the wrong bases in your DNA. Radiation, like from X-rays, can break the DNA strands completely. Chemicals, for example in cigarette smoke and pollution, can also change DNA. Even viruses can cause mutations by inserting their own genes into your DNA. There are also errors during replication, like slip-strand mispairing, where the DNA being copied gets misaligned, especially in repetitive sequences. This can cause insertions or deletions. Thankfully, our cells have repair systems that catch most of their mistakes, but not all.
The external environment can exert powerful effects on the expression of an organism's genes. External factors can include things such as the kinds of pollutants to which organisms are exposed, the amount of daylight they receive, as well as the color of light, and the temperature of the air that surrounds them. An example of a temperature effect on gene expression occurs in Himalayan rabbits. When they are raised at 30 degrees Celsius, they lack any trace of black pigmentation. But when they are raised at a temperature 10 degrees cooler, black pigmentation develops in their noses, ears, feet, and tails. This is because a key pigmentation gene is only expressed at lower temperatures. An example of the effect of the color or wavelength of light on gene expression is observed in certain kinds of butterflies. For example, when caterpillars belonging to the genus Vanessa are raised under red light, they develop into butterflies that possess intensely colored wings, due to the fact that the particular genes that affect wing color in this way are only expressed under red light. But when caterpillars of the same species of this genus are raised under blue light, they give rise to butterflies with wings that have much paler colors, instead of giving rise to the brightly colored specimens produced by exposure to red light. But external factors don't just affect gene expression in the animal kingdom, they affect it in the plant kingdom too. For example, the relative length of light and darkness each day affects the expression of genes that are needed for flowering to occur, a phenomenon known as photoperiodism. The initiation of flowering due to photoperiodism is complex and involves a number of distinct biochemical stages called signal transduction, in which light-induced changes in photoreceptors in certain of the plant's cells cause new intracellular transduction pathways to open up. This ultimately results in chemical signals being sent to the DNA in the nucleus that cause genes to be expressed that code for proteins involved in flowering. So, as can be seen, a very intimate interaction exists between an organism's genes and the external environment in which it lives.
Let's bring this full circle. Mutations don't just affect individuals, they affect whole populations and even the future of species. There are three basic types of mutation effects. Most mutations are neutral, meaning they don't change anything in a meaningful way. These might change a codon, but not the amino acid that it codes for. Some mutations are harmful. These are the ones that can cause disease or make it harder to survive or reproduce. Often, these get removed from the population over time through natural selection. But occasionally and rarely, mutations are beneficial. These help the organism survive better or reproduce more. For example, the ability for adults to digest milk, which is a trait called lactase persistence, is a beneficial mutation that spread in populations that raise dairy animals. Beneficial mutations are rare, but they're the key to how evolution works. Over thousands or even millions of years, small, helpful changes build up and shape the diversity of life that we see on Earth today. So, to wrap up, the major points to remember are: mutations are natural. They happen all the time, often from copying errors or environmental damage. Most of the time, our cells fix them, but when they don't, the change becomes part of the DNA. Next, gene expression is flexible. Our genes don't all turn on at once. Whether they're active depends on things like hormones, diet, temperature, and other environmental factors. This helps organisms adapt to their surroundings. And lastly, mutations fuel evolution. Even though many mutations don't do much, some create advantages. And over time, these helpful changes can spread and lead to new traits and even new species. So, when you think about mutations, don't just think about what goes wrong. Also think about how they drive change and innovation in life. At this point, you should be able to generate a full and appropriate response to today's essential question. What are the causes, types, and effects of mutations?